Hi, Miss Nikki here. Welcome to Chapter 26. We're going to cover this first part of digestion up into the small intestines. So I like these two images side by side. We can see the anatomy here. We can see the oral cavity. We know food's going to move into the mouth. We're going to chew it up. We're going to swallow it. It's going to go through the pharynx, esophagus, come into the stomach here. Then it's going to travel through the three sections of the small intestines. We have the duodenum, we have the jejunum, we have the ileum. Then we're going to dump into the cecum here of the large intestine. We do ascending, transverse, descending. We have that squiggle or sigmoid colon, and then come out the anal canal or rectum, anal canal, anus. Over here, they're really breaking down all of the actions or mechanisms that are happening with digestion. So I would kind of think along these lines a little bit as you're going through the flow of food. So here they're also showing you the alimentary canal. So the tube within your body that's processing and breaking down food, absorbing nutrients, and deciding what's going to be excreted as waste or feces. So alimentary canal from the mouth to the anus, it is a continuous tube. When we look at these mechanisms, you can see ingestion, you're going to put the food in your mouth, you're going to mechanically break it down. So that's the chewing and the churning. You're going to move it or do propulsion. You're going to digest, you're going to be adding chemicals. So here we really have chemical digestion. Then we're going to do absorption here, and then for large intestine, we're going to reabsorb water, reabsorb some vitamins, electrolytes, and then the remainder comes out as feces. So we break down the digestive system into two categories. You can see the gastrointestinal tract. This is really what I just mentioned, the alimentary canal. This is more what they use. You can see the gastrointestinal tract is the oral cavity, the pharynx, the esophagus, the stomach, the small and large intestines, and the anus, and that these are all going to be lined with a mucous membrane. The second category are the accessory digestive organs. So we'll see those on the next slide. And remember, there's always a lumen or an opening, and we're going to see the same pattern that we've been seeing repeatedly. So here's our tube system. We're going to have the lumen in the middle. We're going to have the first layer, which are going to be some sort of mucus cells, right, or mucosal membrane. Then we have that submucosal, then we have muscular, and then we have serous or a fibrous, something adventitia, right? So these accessory digestive organs, these are going to be the salivary glands, the liver, the pancreas, the teeth, the tongue, and the gallbladder. So the gallbladder is going to concentrate and store bile, and the bile is made in the liver. Again, we have the six main functions. We're just kind of breaking it down a little bit more than that first image that we saw. So ingestion, you're going to take either solids or liquid material into the oral cavity. And as soon as it enters the oral cavity, you're going to start digesting it. So we're going to see that in our saliva, we're going to have digestive enzymes like amylase. Then we're going to have to move this material, right? So to swallow, that's going to be voluntary. And then we're going to have involuntary muscle contractions. Think of moving through the pharynx and the esophagus. We're going to have secretion happening. So we're going to see digestive enzymes, acids like hydrochloric acid, and bile dumped onto the food that we've consumed. In digestion, here's that breakdown. Mechanical, so it's some sort of physical breakdown, chewing, mixing, and then chemical digestion where we're going to add enzymes to try to break those bonds down. So we need these smaller molecules in order to do the next step, which is absorption. So now we've taken these digested molecules, we're going to reabsorb electrolytes and vitamins and water, and the smaller molecules that we broke down through both mechanical and chemical digestion, and we're going to move it from the internal part of the digestive tract into the blood or into the lymphatic vessels. And then we have to do elimination. Get rid of anything that's not absorbed. Here are those tunicas or layers. Again, we're repeating the same pattern kind of over and over again. So here's the lumen or the opening. You can see that this mucosal layer in the digestive system is a little bit different in that we have these really thick microvilli. So we're going to see this mucosal layer that has specialized cells for absorbing food. We're going to see submucosal layer. Again, this is always some sort of connective tissue. We're going to see muscularis because we have to have smooth muscle in order to do that peristalsis or alternating contractions. You can also see the muscle layers here. You see how they run different directions. This one is east to west. This one is north to south. And then we've got this outer serous membrane layer. And you can see that there's lots of blood vessels and lymphatic drainage coming into that tissue. The gastrointestinal tract wall mucosal layer is a little bit different because we're going to have simple columnar epithelium that's going to secrete mucus. We're also going to have portions that have non-keratinized stratified squamous epithelium. So think about as you're swallowing food, it's kind of scraping along the sides. This is the lumen. And so you have to offer some sort of abrasion protection. And there are cells being scraped off in your food. Then there's this underlying lamina. And this area is going to have areolar, connective tissue, blood vessels, nerves. And then you have this thin layer of muscularis. And contractions of this area are going to facilitate the release of the secretions into the lumen. So it's a very strange kind of setup where we have two different types of epithelium. We have connective tissue, and then we're going to have this really thin muscular layer that as the food is moving through, it stimulates the secretion of the mucus. In the submucosal layer, we're going to have areolar and dense connective tissue. Areolar gives room for those blood vessels, lymphatic vessels, and nerves. There are nerves and ganglia innervating the smooth muscle and the glands to allow contraction and secretion of the mucus. If you remember, we talked briefly about MALT when we talked about lymphatic tissue. So to prevent large microbes from crossing over the GI tract, we have these lymphatic nodules called Pryor's patches. Remember, anything that you consume probably has bacteria, fungal spores, and no matter how clean it is from the time it's grown to the time it's processed, to the time you cook it, to the time you eat it, it sits on a plate or a fork, you are consuming microbes, so we need to have a defense for that. This muscularis layer, this is smooth muscle. If I say smooth muscle, hopefully we're thinking involuntary control. There's an outer layer. That was kind of what I pointed out, that some are going lengthwise and some are going widthwise. This submucosal plexus and this myenteric plexus were mentioned in the previous slides. Together, it's called the enteric nervous system. So it's going to have some control over secretions, and then it's also going to have some control over the movement of materials through the GI tract. So it's composed of both sensory and motor neurons. Some people think of it as a separate nervous system because it is outside of the central nervous system. Think of it as kind of embedded nervous system in the GI tract. Kind of like we had pacemaker cells in the heart, you have this enteric nervous system that is controlling the secretion of mucus and the alternating contractions like peristalsis through sections of the GI tract. Right now, we're kind of on the mix and propel portion of the GI tract. So the mouth, the pharynx, the esophagus, we're going to have mixing. So this kind of sloshing back and forth motion, and then the blending of the materials with the secretions. So you have mucus being dumped in the saliva. We're going to learn that there are digestive enzymes being dumped on the food, and you're pushing the food into the pharynx, and then you're doing this back and forth mixing as well as alternating contractions, peristalsis. So propulsion, this peristalsis is kind of pushing the material in one direction through the GI tract. So sequential, so alternating contractions of the muscularis, and the wall will kind of move wave-like. I think the image does a much better job of explaining the difference between mixing and propulsion. So here you can see the mixing part. Mixing, you're sloshing the material back and forth as it's moving. And the peristalsis, you have contraction, push. Then you have contraction, push. So they talk about propulsion being directional, where mixing, you have this kind of sloshing back and forth. So adventitia or serosa. So the outer tunic can be either, depending on which organ we're talking about in the GI tract. So adventitia is going to be areolar connective tissue. The serosa is going to be covered in the visceral peritoneum. They just wanted to clarify the overview of the regulation. So we know we have enteric nervous system embedded. These are sensory and motor neurons. These are coordinating smooth muscle mixing and propulsion. And then they are controlling the glands of the GI tract, so secretions. And then we also have our autonomic nervous system. So we have parasympathetic, rest and digest, that's going to increase GI activity, and then sympathetic, fight or flight, which is going to decrease GI tract activity. Peritoneum, by now we should know this. Peritoneum, if it's the parietal peritoneum, it's the serous membrane layer that touches the cavity wall. In this case, we're talking about abdominal. If it's a visceral peritoneum, this is the serous membrane that is directly covering the internal organ. And the peritoneal cavity would be the space between those two layers. We're going to have serous fluid, and it allows those abdominal organs to kind of move. Remember, there's a lot of mixing and a lot of muscle contraction to move food through the GI tract. So first off, we're talking about the abdominal pelvic cavity. We're seeing the parietal, so this is the outer serous membrane layer that's touching the cavity wall. And then we see the visceral peritoneum, and that is the inner serous membrane layer that's actually touching the organ. So this would be the peritoneal cavity. It would be filled with peritoneal fluid. Remember, we had some items that were outside of the peritoneal cavity. So these were those retro. I'm kind of drawing kidneys. I'm going to abbreviate. So there are some organs that lay outside of the peritoneum and some that are inside. The ones that are inside, we're going to see a folding of the parietal peritoneum and the visceral peritoneum, and they kind of offer stability. They're kind of roping off these digestive organs in this really large abdominal or abdominopelvic cavity. And the term that we use is called mesentery. So here, mesentery, double layer of the peritoneum. It's going to offer support. It's going to stabilize the organs that are inside the peritoneal cavity. We're going to see blood, vessels, lymphatic vessels, nerves sandwiched between the folds. We also see this, or name it, greater omentum and lesser omentum. So you're going to have, and I have an image coming up in just a second, you're going to have this fatty apron or this adipose tissue that's covering most of the abdominal organs. Usually we can see that it's covering the majority of the small intestines, and it's acting to kind of keep the abdominal organs kind of in their space. And then we have this lesser omentum. This attaches from the stomach to the proximal end of the duodenum of the liver. Duodenum is one part of the gastrointestinal tract that is outside of it, is retroperitoneal. There's a cute mnemonic in your book, if you want to go look it up, SAD Pucker, that goes through all of the sections of all of the organs that are outside of the peritoneal. So all of the ones that are retroperitoneal. So supraadrenal glands, aorta, duodenum, pancreas, urinary bladder, ureters, colon, kidneys, esophagus, rectum. Here's a cadaver image showing you the greater omentum. You can see it really does act like a fat drape or a fat apron. And then the greater omentum on the model, they cut it away so that you could see the small intestines. But if you see the purple guy in lab, he does have a section of greater omentum. And then we said there's another section, the lesser omentum, is going to attach the stomach and the duodenum. And then we also have this liver attachment here. Some people consider that part of mesentery. And then there's also the mesocolon. So on the back side, part of the transverse colon, and where the sigmoid colon is, there's another attachment. So some of this mesentery is kind of all over the place, keeping all of these abdominal organs kind of stable within this abdominal cavity and inside the peritoneal cavity. So peritonitis, you could imagine if you had inflammation of the peritoneum, just like we've seen it in pleura or cardial. So if you remember, we talked about the visceral and parietal pericardium inflammation, and then we talked about visceral and parietal pleura inflammation. So it can happen in the peritoneum as well. This would be abdominal pain. It's usually some sort of GI tract perforation, so a tear somewhere in the GI tract. It allows the contents to come in contact with the peritoneum. Ulcer, ruptured appendix, gunshot wound, surgery. So now we're going to talk about the upper GI tract and also talk about the accessory organs. So here we have oral cavity, that is upper GI tract. Salivary glands, those are the accessory structures. So we're going to see mechanical digestion because we're going to put the food in our mouth and we're going to start chewing it. Then we're also going to see chemical digestion because the salivary glands are going to dump saliva, and that saliva has amylase. And I kind of mentioned this already. We're starting the starch or complex carb breakdown. It's going to mix with the food. So we've got mucus and food and saliva, and that's going to form a bolus. The bolus is going to be pushed into the pharynx. The tongue is going to initiate swallowing, and then eventually the pharynx is going to take over with propulsion. Also, we have to have mucus secreted to facilitate the swallowing. We do not want to put food into this tube system that doesn't have mucus on it. It'll be really hard to swallow. I always say, imagine going down a water slide with no water. So we've taken that bolus from the oral cavity, we've pushed it into the pharynx. Now I need to get it into the stomach. The esophagus is going to lead from the pharynx to the stomach. We're going to have mucus secretions lubricating the movement or the propulsion of this bolus into the stomach. That bolus, once it's in the stomach, is going to mix with gastric secretions, and the stomach is going to do lots of contractions, and you'll see how the muscle fascicles or layers line up in the stomach in just a second. But there's lots of mixing, lots of contraction, and we've dumped gastric secretions on it. So again, you're going to have hydrochloric acid, which is going to be a chemical digestion. And then you're going to have these smooth muscle contractions, which are going to be mechanical digestion. Now we call it chyme. So chyme is, as it leaves the stomach, it's gone from a bolus to chyme. Chyme is dumped into the duodenum, which is still considered part of the upper GI tract. So now we look at the oral cavity, the mouth. There's a vestibule, there's different areas. There's an oral cavity proper. So the vestibule is the entire space between the gums and the lips and the cheek. The oral cavity proper is central to the teeth, leads to the oral pharynx, and then we have our cheeks. If you remember, your buccinator or buccinator muscles allow you to hold the material while you're chewing. We have lips, we have palate, we have soft palate, hard palate, uvula. We already talked about these when we discussed respiratory. We have the tongue. So the tongue, if you remember from 201, we have lots of projections or papillae. We have taste buds. The tongue is going to manipulate and mix the materials. So the tongue is kind of moving the food around. It's getting the saliva on it, the mucus. You're breaking it apart with your teeth. And the tongue is the initial mechanism or initial organ that allows for swallowing. And then your pharynx will take over with the propulsion. So here we have our hard palate. So imagine you're pushing the food to the roof of your mouth, right? So you're smashing it against the bone, the maxilla, the hard palate, or your palatine bone. Remember, the first half is maxilla and the back half is palatine. So you're smashing the food, chewing the food. You're doing mechanical digestion with the tongue. The soft palate, we have the uvula. Remember that when the tongue pushes the food back to the oropharynx, that uvula is going to hit the back of the pharynx and stop you from pushing food into your nasopharynx. So we have food in, we chew it, the tongue pushes it back to the oropharynx, and now as it's traveling the pharynx to the esophagus, that's where we have those alternating contractions. Remember, we do have the epiglottis, the flap that's going to close so that you do not push food into your trachea or airway, or larynx, then trachea. What's lining the oral cavity? You have stratified squamous epithelium. Hopefully by now we see stratified squamous epithelium and we're thinking some sort of abrasion protection. It is mostly non-keratinized. We do have some keratinized stratified squamous epithelium, the lining of the lips, portions of the tongue, and part of the hard palate. Salivary glands, we have to go through 3 of them. We have the parotid. This is the largest, but it doesn't produce the majority of saliva, but it is the large salivary gland on the side by the ear. It's going to push the saliva into the oral cavity through the parotid duct. This is also the gland that gets infected that we would call mumps. You can see here submandibular, so this would be the salivary gland below the mandible. This produces the majority of the saliva. Then we have sublingual. This would be underneath the tongue or inferior to the tongue, and a little bit of saliva. So what is saliva? We secrete about 1 liter a day, especially at mealtime. Most of it is water. There are some solutes in there. There is salivary amylase to break down those complex carbs. We're going to have mucin when mixed with water becomes mucus, and then we're going to have lysozymes. So remember, lysozymes antibacterial, they can inhibit bacterial growth. Saliva also helps keep your teeth clean. People that have issues with decreased saliva production have issues with cavities, bacteria in their mouth. So that salivary amylase is going to initiate the chemical breakdown of starch complex carbs. We can also see that parasympathetic stimulation would cause an increase in salivary secretion, where sympathetic is going to result in more viscous saliva, so less watery, thicker saliva. Here's a visual. You can see the parotid. You can see the parotid duct. Sometimes the parotid duct can get clogged, and then that can cause inflammation in the parotid salivary gland. You can see the sublingual underneath the tongue, and then the submandibular, and we have these on both sides. Now we move on to talk about mechanical digestion or mastication, the chewing. So this is happening in the oral cavity. We have our teeth that are chewing and tearing the food apart. We're smashing it to the roof of our mouth, our hard palate with our tongue. We're going to dump saliva on it, right? So we're doing this mechanical, trying to take bulk and turn it into smaller molecules. We're also dumping enzymes like amylase on it from our saliva. So certain medications can be placed underneath the tongue, and they can pass through the epithelium by simple diffusion. So you might know somebody that takes nitroglycerin tablets. They do have, I think it's Maxalt for migraines. So there are some medications that can be easily absorbed underneath the tongue. Here are the deciduous teeth. You might want to think of them as baby teeth. Some people call them milk teeth. You can see that we have incisors and we have canines, and there's a first and a second molar. These teeth are going to fall out and hopefully be replaced with permanent teeth. In some cases, the permanent tooth isn't there when the baby tooth falls out. The permanent tooth is just not in the maxilla to be pushed down. So we can see we have central incisors, lateral, we have canines, we have premolars, we have molars. And these would be considered those wisdom teeth that come in later in life. Now we move on to the pharynx and the esophagus. So once we've masticated the food, we've chewed the food up, we've dumped salivary amylase on it, we're pushing it with our tongue back into the pharynx. Now we're going to move it pharynx to esophagus. So we have skeletal muscles that are going to aid in the swallowing. This area is going to be lined with non-keratinized stratified squamous epithelium, abrasion protection, but we're not making that lipid protein keratin. The esophagus is normally collapsed. The section or the opening of the diaphragm where the esophagus enters, and then we'd have stomach here. That opening is called the esophageal hiatus. So this would be diaphragm. So let's follow the flow of food. So the food is put into the oral cavity. We're going to use the tongue to mash the food. We're going to use the teeth to tear the food. We're going to dump saliva, start the complex carb digestion, and then the tongue is going to push the food back into the pharynx. So food goes from the oral cavity, then it goes to the oropharynx, then to the laryngeo or laryngopharynx, and then it's going to move down the esophagus, travel through the diaphragm at that esophageal hiatus, and now we've finally gotten to the stomach. And we're going to go through all the parts of the stomach. So the esophagus, again, we have the mucosal layer, we have the submucosal, we have the muscularis, and we have adventitia. So the actual process of swallowing is called deglutition. So the voluntary phase is controlled by your cerebral cortex, your conscious control. The bolus and the saliva are going to mix, and you're going to push the bolus into the oropharynx with the tongue. Then you have this involuntary reflex. So the sensory input, there's something in the pharynx, goes to the swallowing center, which is in the medulla oblongata, and then we relay those effectors to the muscles to actually start that reflex or alternating contractions to propel the food down the pharynx. So visual here, you can see voluntary phase, the tongue is pushing the food, and we push it back into the oropharynx, and now the involuntary phase, the pharyngeal phase, is going to take over, and we're going to move that bolus through the pharynx and down the esophagus to the stomach. You can have issues where you can have inflammation of the esophagus. You can have reflux disease, so from the stomach to the esophagus, imagine heartburn, right? Or, we haven't talked about it really yet, but the hydrochloric acid from the stomach getting pushed up into the esophagus. That would probably not feel really good, right? So heartburn. There's also something called a hiatal hernia. So remember how we had the diaphragm and we had that opening where the esophagus travels through the diaphragm? Well, sometimes portions of the esophagus or the stomach can push through that esophageal hiatus, and that's called a hiatal hernia. Now we move to the stomach. So the stomach is going to do both chemical and mechanical digestion. We saw this already in the oral cavity. So we dumped the saliva and we had the teeth and the tongue mashing. So we're just kind of continuing that on in the stomach. So oral cavity does chemical and mechanical. Then you propel it down the pharynx and the esophagus. Now when we get to the stomach, we're going to do this again, but you can see that we're going to start digesting proteins and fats. So food material is going to spend about 4 hours, 2 to 6, depending, in the stomach, and then we're going to push it to the small intestines, and that's where absorption really happens. We do see some limited absorption in the stomach. Really, this is just a holding bag. We're dumping chemicals on the bolus of food. We're churning it, dumping chemicals, churning it. The entire point of this is to turn the bolus into chyme. So we have more bulk. We started digestion, and what are we trying to turn it into? Really small molecules that we can actually absorb. There's a greater curve, a lesser curve. There's a cardiac sphincter. They call the area or the region cardia, and then we're going to see an opening or the cardiac orifice where the cardia and the esophagus meet, and then the opening and closing of that region. Then we're going to see the fundus, the body, and the pylorus region, and a pyloric sphincter. Inside the stomach, you're going to see rugae. These are gastric folds. They're on the lining of the stomach when the stomach is empty, and they allow for expansion. They return to normal when empty. Remember, we also have greater and lesser omentum. So in bold are all of the regions. So we see the bolus of food moves from the esophagus into the stomach. It hits the cardia region, then the rounded fundus, then the body, then the end section or most distal section, the pylorus. So if I said name the region, these are the terms: cardia, fundus, body, pylorus. If we ask for specifics, like name this sphincter, here they did use the term orifice, cardia orifice, also called, or in that area there'd also be the cardiac sphincter. So I think there's a little discrepancy between the textbook and our lab book. I don't believe it says cardia orifice, I believe it says cardiac sphincter. Here you can see pyloric sphincter, the muscles here that would close off the chyme from moving from the pylorus into the duodenum, which is part of the small intestines. And then the opening is the pyloric orifice. Now we need to look at the histology and look more at the gastric glands. So we're going to see that these gastric glands extend deep into the mucosa. They're partially surrounded by muscularis mucosa, and those help expel those gastric secretions. We're going to see that there are 3 layers of smooth muscle in the stomach. We're also going to see the outer layer, the serosal layer this time. So it is visceral peritoneum that's touching the outer wall of the stomach. So here are the tunicas, right? Here's all of the mucosa. And remember, this is a little strange because we have this muscularis mucosa, which is going to aid in the secretion of the cells of the mucosal layer. Then we have this submucosal layer. Again, lots of connective tissue, areolar. We're going to see blood vessels and nerves and lymphatics. Then we're going to see 3 layers of muscles. And then we're going to see the outer layer, which is the serosa, which is the visceral peritoneum. Peritoneum. Something that I didn't point out before, if you go back and look at the anatomy of the stomach, you'll see that the 3 different layers of the muscle wall of the stomach, that all the fascicles run in different arrangements. So you'll have a north to south, and east to west, and then you'll have an oblique. So here's the gastric pit is really the top part, and the gastric gland is the bottom part. So in the top section, we're going to see 2 cells that secrete mucus. In the bottom section here, gastric gland, we're going to see parietal cells, chief cells, and then enteroendocrine cells that are going to secrete hormones. There are lots of cells, 5 to be specific, that are going to secrete some sort of substance. So 4 are going to produce gastric juice, and the 5th is going to secrete hormones. So we have the surface mucosal cells. These are the first ones as we're going into the pit. So these surface mucosal cells are going to secrete an alkaline product containing mucin. Again, mixed with water, it becomes mucus. So this is going to help prevent the stomach lining from being digested by the hydrochloric acid that's going to be used to break the food down. So these mucus neck cells, these are the second set of cells. So here is the gastric pit area. We had the first set of mucus cells. Now we have a second set of mucus cells. They are also going to help protect the stomach lining from abrasion and injury from some of these gastric cells that are going to secrete things like hydrochloric acid. So let's look at the parietal cell in more detail. It's going to secrete 2 substances. One is intrinsic factor. You need intrinsic factor to absorb B12, and you need B12 to make red blood cells or erythrocytes. The second is hydrochloric acid. So the parietal cells are going to aid in the formation of hydrochloric acid, and this is responsible for the low pH of the stomach, pH of around 2. So the hydrochloric acid is going to help break down plant cell walls, animal connective tissue, denature proteins, remember proteins, function at specific temperatures and pH, and they can be broken down. Chemical digestion is going to happen. We're also going to activate pepsinogen into the active form of pepsin. So we've seen kind of this inactive enzyme has to be activated. We saw it with the clotting factors, right? Intrinsic, extrinsic. And this also kills a large majority of any of the microorganisms that would be entering the stomach mixed in that bolus with the food that you consumed. So I have a kind of more detailed picture here of the parietal cells just because I want you to see this is kind of interesting. I'm not going to ask you details about this, but the parietal cells cannot store hydrochloric acid. I said they facilitate in dumping hydrochloric acid into the stomach opening. If the parietal cells made it and stored it, they would be degraded by it. They would break down. But they're showing you this equation, which should look familiar. So this is a cell. This cell undergoes cellular respiration. So does it release carbon dioxide and water? Yes. Does that carbon dioxide and water turn into carbonic acid? Is it then broken down into bicarbonate and hydrogen ions? Yes, we've seen this before in the bicarbonate buffer system when we did respiratory. So the bicarbonate gets shifted into the blood capillary, and that causes the backward shift of chloride into the stomach lumen. Then we're going to see these hydrogen ions pumped into the stomach lumen, combine with the chloride and form hydrochloric acid. Chief cells are the most numerous. These are going to have plasminogen. So this is the inactive precursor of pepsin. Pepsin must be in inactive form to prevent the destruction of the chief cells. So we have to store it again as pepsinogen, and we don't want to activate it because pepsin will start breaking down the proteins inside the actual chief cells. So pepsinogen is activated by hydrochloric acid to become the active form of pepsin, and pepsin is going to denature proteins. We're also going to see the chief cells release gastric lipase. So this is not a huge role in fat digestion, but we kind of start the fat process or fat breakdown process. For these G cells of the stomach, I really just want you to think of them as enteroendocrine or hormone-producing cells. And these are going to secrete things like gastrin, which gastrin can increase stomach secretions, increase motility or the movement and mixing of food. And then other types of hormones like somatostatin, which can decrease the secretions and motility. I'm not going to ask you questions about this other than, do you know that endocrine hormones are released from stomach G cells, which are in those gastric glands? So this somatostatin also has some connections to the pituitary, also has some connections to the pancreas, also has some connections in memory. So kind of interesting, you could think about the stomach filling, you could think about these hormones, you could think how these hormones and your stomach filling could contribute to the feeling of being full, talking to your brain, right? So this is why, I think there's a lot more to this than what we're going to talk about in class, but you could see how, when they say there's always a lag between your gut and your brain. So kind of take a second as you're filling your stomach, stop eating before you're actually full, because these hormones have to talk from the stomach to the brain, and there is a delayed effect there. Kind of just a follow-up summary here. We have the two cells that are secreting mucus to kind of make the internal lining more alkaline. So we do have all of this craziness going on with hydrochloric acid and denaturing proteins. So we're going to secrete a whole bunch of mucus, kind of line the internal or stomach lumen and keep that more alkaline so we don't start digesting our actual gastric glands. Then we have the parietal cells. The parietal cells are going to facilitate the secretion of hydrochloric acid. Then we have the chief cells, which are going to take or release pepsinogen inactive, which then gets activated by the hydrochloric acid, which then turns into the active form, which is pepsin, and we start seeing a breakdown of proteins in the stomach. So the hydrochloric acid plus the pepsin are both going to aid in protein digestion. We also mentioned there's a little bit of lipase helping with fat digestion. We also have to think about that intrinsic factor from that parietal cell. So that intrinsic factor is going to allow us to absorption of vitamin B12, which then allows us to make more erythrocytes or red blood cells. And lastly, we have these G cells, which are releasing hormones into the bloodstream to tell us we're full or tell us to increase or decrease digestion. So we're going to see two primary functions. We're going to mix the bolus of food to form something called chyme. So think of it as bolus is what enters the stomach, and chyme is what leaves the stomach. So we're going to have mixing, mechanical digestion. We're going to change the bolus into chyme. We're going to reduce the size. Again, we're going from bulk to more small molecules so we can absorb them when we get to the small intestines. Gastric emptying. So we're going to move the chyme from the stomach into the duodenum. As it moves, it's going to move into the pylorus, the most distal part of the stomach, and that the force pressing on the pyloric sphincter causes it to open, and the chyme is going to slosh out some of it, and then the sphincter is going to close. And so we're going to have this sloshing back and forth, or this reverse flow of some of the contents back towards the stomach. So retropulsion. So the bolus of food is what enters. We dump all the gastric secretions on it. We're breaking it from larger molecules into smaller. As the chyme hits this pyloric region, it puts pressure on the pyloric sphincter. Pyloric sphincter opens. We push some of the chyme into the duodenum, and then some of it backflows. So it's this constant churning and mixing and digestion from the hydrochloric acid and the pepsin. So even though a lot of people think about the stomach as just a holding area for partially digested food, there is a lot going on here, right? We're dumping chemicals on it, we're churning, we're mixing, we're doing that retropulsion, that back and forth. There are pacemaker cells in the stomach that are constantly depolarizing. They don't have that long resting membrane potential, and they establish the rhythm of muscular contraction. So that's going to spread through the smooth muscles. That's going to regulate the force of contraction and the secretion of the glands. So again, I want you to see kind of how the brain and the stomach are working together, and see kind of some of the pathways of these sensory and motor outputs. I don't test on the regulation of the digestive processes. We're not going to go through these different reflexes. I kind of just want you to get an idea of how complicated this is. So we have this cephalic reflex. So you're thinking about food, you see food, you're getting ready. So you have some sort of special sense receptor that's activated. You have sensory input to your cerebral cortex. You're sending a message down the brainstem via the vagus nerve, and that's going to cause your stomach to increase secretions and start rumbling. So if you've ever seen food and you're really hungry, you feel your stomach rumbling. So that is that initial reflex. Then we have the gastric reflex. So once you've actually consumed the food and we have food in the stomach, we're going to have chemoreceptors and baroreceptors. Remember, baro detects stretch and chemoreceptors chemicals. Well, remember, if you have a really high pH, that's due to those parietal cells facilitating that hydrochloric acid, and that can all be detected, and that is sent back to the brain, so the medulla, and then we're going to have increased forcefulness of contractions and secretions. We're also going to have the hormone gastrin secreted to also stimulate the force of contractions and secretions. Then we have the intestinal reflex. So once you move the food into the duodenum, that's going to say, okay, we've emptied the stomach, we've now moved the food into the small intestines, now we can go ahead and decrease the forcefulness of contractions and secretions. And there are hormones released to facilitate those actions. Gastric bypass, Sometimes I've heard of it mentioned as gastric sleeve. They used to actually put a sleeve over the stomach to stop it from expanding fully. Now they actually cut out or surgically remove part of the stomach. I don't know if they do the gastric sleeve anymore. I haven't read up on it lately. But basically, less nutrients are going to be absorbed, reduced appetite, altered response to hormones like insulin. And most of the time, if somebody is getting a gastric bypass, it's because they have so many other issues that are life-threatening. So they could have high blood pressure, they could have diabetes, they could be having cardiac issues, they could be having edema, swelling in the legs. So there's lots of reasons that we would want to do this, but pay attention to the fact that less nutrients are being absorbed. Remember, the whole point is we're taking the bulk and we're breaking it down into the small molecules. Well, if you have turned the stomach from this into this, you're not going to be able to reduce the bulk food down into small enough molecules to be able to absorb. So you're going to have limited absorption. So your diet will have to be very specific. A lot of people that have gastric bypass, they have to make sure that they are taking enough calcium, vitamins in general. You would also have to pay attention to how hormones are going to affect how much glucose you're taking in, how much fat you're taking in. So limited diet, and not only that, you can only consume so much because you've actually removed part of the stomach. You could imagine that erosion of portions of the stomach or duodenum could happen if you had some issues. You could have the hydrochloric acid and the pepsin that are actually eroding away part of the stomach or duodenum. These ulcers could then lead to perforation. So remember, the outer layer of the stomach is a serous membrane, and we talked about visceral peritoneum is that outer lining. What happens if that is perforated and now we have hydrochloric acid and pepsin leaking into the peritoneal cavity in the abdominal cavity? And there is a bacteria that's known to cause erosion of the gastric lining. Also, we could talk about vomiting. So this is the rapid expulsion of gastric contents through the oral cavity. So instead of leading to the duodenum, the vomiting center can be triggered by trauma, motion sickness, infection, toxicity, like food poisoning, right? So the medulla oblongata triggers the vomiting center, and instead of pushing items down, doing the peristalsis down, instead you start peristalsis going backwards, and you're going to have that retropropulsion, and you're going to take the stomach contents and push them out the oral cavity. Hopefully your nasal passage will close your uvula and your glottis. We do not want the food to go to the glottis because that would lead to the trachea. We do not want to aspirate the contents if we're trying to get it out of the body. So these contents are forced into the esophagus. This is why if somebody's throwing up, you're supposed to turn them on the side so that they can't choke on their own vomit, right? Or they can't aspirate the vomit. You could also think that this is going to affect the overall blood pH. So if you're removing the acid that would normally be deposited in your blood capillary, so you remove the acid, your pH is going to become more alkaline. So remember how we talked about metabolic alkalosis. So metabolic alkalosis, one of the reasons could be vomiting. All right, well, that's it for part 1. I'll talk to you in part 2. Bye.